continuation of a lecture series on cell cycle and cell division. We have reached the third and the last of this series. Here today we will be talking about the type of cell division, that is meiosis. Now, meiosis, as we have already tried to understand what is meiosis. Meiosis is a type of cell division which is mainly responsible for formation of the gametes during reproduction of any organism, sexual reproduction of any organism. Now, meiosis is a little lengthy process, and as such the entire process of meiosis is divided into two broad phases. They are meiosis 1 and meiosis 2. As you can see here, it's meiosis 1 and meiosis 2. It is divided into two broad phases. Now in meiosis 1, meiosis 1 is further divided into prophase 1, metaphase 1, anaphase 1, telophase 1, followed by cytokinesis, where we will get two cells, or these cells sometimes they remain attached to each other even, then we call them as dyads. They are also known as dyads, in this case two cells. Now these two cells will then undergo the second phase, that is meiosis 2. Even this meiosis 2 can be divided into prophase 2, metaphase 2, anaphase 2 and telophase 2. Then finally again cytokinesis takes place, giving us this 4 new daughter cells. 4 daughter cells at the end of the entire process of meiosis. These 4 cells which are formed, these 4 daughter cells which are formed, each of them will be having half the number of chromosomes to the original parent cell. So this type of division is also known as reduction division. Now the cell, as like in mitosis, the cell first undergoes the period of interphase. Interphase, when it will the DNA will duplicate, forming the chromatids, and then followed by which the centromeres also are also formed in case of animal cells. So that is the interphase. Then it starts with prophase 1. In prophase 1, what will happen? The chromatids will be more distinct. Now this prophase 1 is further divided in case of meiosis 1 into 5 phases. They are liptotin, zygotin, pekhitin, diplotin and diakinesis. 5 phases, it is 5 steps it is divided, because the prophase 1 itself is very long and lengthy phase. Then it is followed by metaphase 1, anaphase 1, telophase 1 and then meiosis 2 starts. Now we will talk about prophase 1 more in details. Prophase 1 more in details, and then we will again come back to metaphase 1, anaphase and the rest of the divisions. First, leptotin. Leptotin is what? Leptotin is the first step in case of prophase 1. In prophase 1, the first it moves through leptotin. Here prophase begins. Chromosomes start to condense. The chromatin, which was present during the interphase, it's just a long thread inside the nucleus. Now what will happen? It will gradually start to condense. They will start to condense, and then the pairing of homologous chromosomes. Now you must be thinking, what is this homologous chromosome? Now we all know that in case of diploid organisms, the cells, they have pairs of chromosomes, 2 sets of chromosomes. Like we humans, we have 46 chromosomes. That means 2 sets, 2 haploid sets, 23 plus 23. Now when we say 23 plus 23, that means we are talking about the pairs. Now the first of each set, say chromosome number 1 from the first set, chromosome number 1 from the second set, they will form a pair. That is called as homologous pair. Likewise, the second from each set, they are called, they will also form a pair. That will be the second pair, second homologous pair, third homologous pair, fourth homologous pair. So that way, in diploid organisms, chromosomes always exist in pairs. In diploid cells, they always exist in pairs, and these pairs are known as homologous chromosomes. Why are we telling about it? Because in prophase 1 of meiosis, what will happen? The homologous pairs, they come together, as you can see here. Okay, so here what is happening? The homologous pairs are coming together. Now here, blue and red, here they have shown 4 chromosomes. As you can see here, they are showing here 4 chromosomes. Now these 4 chromosomes, that is blue, they belong to one set, red belong to one set. So one blue, one red, they will form a pair. The other blue and the other red, they will form a pair. Okay, so here this way, this is one homologous pair. So prophase begins in leptotene, where the chromosomes start to condense and gradually they start pairing. So here you can see they have started pairing. The pairing starts when we call it as zygotene, and then synapsis begins. Now what is this synapsis? In this synapsis, what will happen? They will get attached at certain points. Okay, the homologous chromosomes will the two chromosomes lie exactly opposite to each other, and the same characters they attach. They lie exactly end to end. End to end, they lie against each other, and then get attached, not permanently, temporarily, they form a complex. Okay, they form a complex which we call as synaptonemal complex. So this is known as synapsis. The process of association of the chromosomes is known as synapsis. Then it moves over to pectine. In pectine, what will happen? The chromosomes, as they have paired up, they have formed the synaptonemal complex. Now, also, you are familiar that each chromosome is made up of 2 chromatids. What are those 2 chromatids? You have to recall back. Okay, what are those 2 chromatids? The duplicated DNA. The duplicated DNA, they form the chromatids. So now, what do we see? Each chromosome has 2 chromatids. So, a pair which has 2 chromosomes has 4 chromatids. 2 in this blue, 2 in this red. 2 blue, 2 in the red. Okay, I hope you're not confused. So here, always remember, each chromosome is already made up of 2 chromatids, and here pairing has occurred. 2 chromosomes have come together. So obviously there will be 4 chromatids in each pair. This structure is also sometimes called as tetrad. These are 4, that is why tetrad, or sometimes this is also known as a bivalent. It is also called as bivalent. Then wherever they are associated, these temporary associated points, upon them crossing over or exchange of DNA takes place between non-sister chromatids. Now you see here how it is happening. Okay, how it is happening here? You have this. These 2 are the chromatids, these 2 are the chromatids. These 2 blue ones are sister chromatids, and 1 red, 1 blue will be called as non-sister chromatids. Now here they have crossed each other. At this point you can see they have crossed each other. Okay, cross means they have interacted with each other. These events happen in pectine, and this event is known as the process of crossing over. During crossing over, what happens? There is exchange of genes. That is, this one part of blue will come over to red, and this part of red will go over to the blue. So exchange of genes takes place. This is also called as recombination. By the end of pectine, recombination gets completed, leaving the chromosomes linked at the site of the crossing over. So in diplotin, okay, when we have reached diplotin, now what happens in diplotin? Diplotin, you can see the synapses ends. Now here this exchange has taken place. As I told you, this blue, a portion of red has come over here, and in the red, a portion of blue has gone over here. As an example, just a small portion is shown. Otherwise, at many different points, such crossing overs will occur. So here synapses, it ends, the chiasma visible within the bivalve. Now what is this chiasma? The X-shaped structure. This X-shaped structure, this is known as chiasma. This is chiasma. So it is visible. So here synapses ends. That means even the crossing over has ended. Then finally, diakinesis. In diakinesis, that is diakinesis, what will happen? This is terminalization of leptotin. That means the leptotin has almost come to an end. Chromosomes become fully condensed again. The nucleolus degenerates now. By this time, the nucleolus has degenerated. Then the breakdown of the nuclear envelope also happens around this time. Then the mitotic spindle formation will also occur at this time. And finally, what you will get? You will get this. Already you have this recombinants are already present here. So now here, these are the kinetochores attached to the centromeres of each chromosome. So here, the two homologous pairs are still together, but this is in a new form. This was not what was present originally. Originally, you can see this was a complete blue and this was complete red. Now, what has happened here? The exchange has taken place. And so, you have a new combination of chromosomes now. So, that marks the end of prophase 1. Now here, prophase 1, just to make the explanation more elaborate or better, meiosis 1, prophase 1, synapses and crossing over occur. This is the main event here. Homologs align independently. So what will happen? These are the sister chromatids. As I told you, this is a pair of bivalent. There's a pair of bivalent. There's 4. That is why they are called as tetrads also. Each chromosome has 2 chromatids. Each chromosome has 2 chromatids. Then after which it is followed by this synapse formation, exchange, crossing over between non-sister chromatids. So here you can see how the exchange has taken place between this and between this. After exchange, then resulting daughter chromosomes. So you can see these will be the daughter chromosomes later. So this is prophase 1. Now, meiosis, again, 2 stages. Interphase, where does it take place? In the reproductive cells, that is germ cells in the gonads, that is reproductive organs. Prophase 1, synapses for crossing over, we have already discussed. So once synapses has taken place, it moves over to metaphase. In metaphase 1, what will happen? All the homologous chromosomes, they will get arranged in the equatorial plate. Quite similar to the earlier cases of mitosis. So they will get arranged in the central plate. In the meantime, spindle fibers have formed from the centromeres. Then gradually they will be pulling apart of the homologous chromosomes. Now here you have to take note, in mitosis what happened? The chromatids separated. But here what is happening? The homologous chromosomes are together. So to each pole, half of the chromosomes move, and to the other pole, half of the chromosomes move. Okay, that is, but what type of chromosomes? Recombinants. They move to both the poles. This is known as anaphase 1. They get separated, the chromosomes. So what happens? Here, how many were there? There were 2n chromosomes in pairs. Now, one set moves to one pole, the other set moves to the other pole, resulting there will be n chromosomes in one pole and another n in the other pole. So you get a reduced number of chromosomes now. So here that marks telophase 1 end. Now at this stage, there are 2 nuclei. The nuclear membrane again reappears. There are 2 nuclei, when we can also call this particular cell as dyad. It is also referred to as dyad now. So all of these, they are together now. They remain together. It is known as dyad. Now it will move towards, or in some cases they may even separate out through cytokinesis. Then it moves to meiosis 2. In meiosis 2, what will happen? Each of these two independently, the chromatids will reappear. The chromatids, because if you can recall, each chromosome has two chromatids. They are still there. So those two chromatids will now arrange themselves, become condensed, and then arrange themselves in the cytoplasm. Within the cytoplasm, nuclear membrane will again disappear when it is known as prophase 2. After which they will arrange themselves in the central plate, equatorial plate, and then which is metaphase 2. The spindle fibers again are formed, they reappear, and then these chromatids will be pulled apart now. Now the chromatids are pulled apart. Now it's not chromosomes. Now these are the chromatids. So here n chromosomes were there. So again n chromatids, because each chromosome has two chromatids. So they will separate up. Similarly, both the cells it is happening, which is known as anaphase 2. And finally, what will happen? The chromatids, they aggregate together to form the chromosomes, when we will call as telophase 2. So finally, you get 4 cells with 4 nuclei, each having half the number of chromosomes. If you have followed properly, you can see here that meiosis 2 is exactly similar as mitotic type of cell division, but of course the difference remains that here it happens only with haploid set of chromosomes in both the two cells, whereas in mitosis it is diploid chromosomes. So this is about meiosis 1 and meiosis 2. Finally, when we are getting 4 new daughter cells, each of them will behave as the gametes. Now, the significance of meiosis is that it helps in the reproductive process, as also it brings about variation in the organisms through the process of reproduction. Now, where did that variation occur? Here, in prophase 1, when crossing over had taken place, new combination of chromosomes appeared.